everyone, this is Marina, and the title of our article is SARS-CoV-2 Cell Entry Depends on ACE2 and TMPRSS2 and is blocked by a clinically proven protease inhibitor. So this study was conducted in the Stefan Pohlmann lab at the University of Göttingen in Göttingen, Germany. The article is published in Cell, which has an impact factor of 36.216, and the work was supported by grants from BMBF and the German Research Foundation, and none of the authors declared any competing interests for this study. So to summarize the abstract, the SARS coronavirus 2, also referred to as SARS-CoV-2, has presented a global health emergency and a worldwide pandemic. But entry into host cells is dependent on viral spike proteins binding to cellular receptors. So these spike proteins are often referred to as S proteins for spike protein. And understanding which factors are used by SARS-CoV-2 will further our understanding of viral transmission and potential drug targets. And so this study specifically determined that the receptor ACE2 is used for cell entry, and TMPRSS2, which is a serine protease, is used for spike protein priming and helps cleave the spike protein into S1 and S2. And so overall, in general, this article suggests a TMPRSS2 protease inhibitor could be a viable treatment option based on the testing done in this article. So now I'm going to introduce some of the prior knowledge that the authors went into already knowing when conducting this study, and just a brief overview of coronaviruses. So coronaviruses can cause mild to severe respiratory disease in humans. SARS-causing coronaviruses are enveloped viruses containing a single-stranded 5'capped positive RNA that ranges from 26 to 32 kilobases and contains at least 6 open reading frames. And so SARS, which is severe acute respiratory syndrome, emerged in 2002, and that also caused a pandemic at the time. And to date, there are no specific antivirals or vaccines that are used to combat SARS. And this syndrome is caused by SARS-CoV, sometimes also referred to as SARS-CoV-1 or just SARS-1. So keep that in mind while going through this presentation. Sometimes it might be referred to as either SARS-1, SARS-CoV-1, or just SARS-CoV. COVID-19, on the other hand, is coronavirus disease 19, which, as many of us know, emerged in December 2019. And COVID-19 is caused by SARS-CoV-2. So again, throughout the paper, sometimes this is referred to as SARS-CoV-2, sometimes SARS-2, sometimes just coronavirus. But that's just something to keep in mind. SARS-CoV entry into host cells is dependent on the spike protein binding to a specific host cellular receptor, as well as protein priming by host cellular proteases. And this priming process allows for the fusion of viral and cellular membranes. So the SARS spike protein is known to engage ACE2 as its entry receptor, but not all cells express this surface receptor. And TMPRSS2 and a cysteine protease are used for protein priming. And so that's known for SARS-CoV-1. So it's still largely unknown if the SARS-2 spike protein employs ACE2 and TMPRSS2 or CATBL for host cell entry the same way SARS does. And so the overall hypothesis is that SARS-2S also utilizes that ACE2 and TMPRSS2 for host cell entry and infection. And so throughout the presentation, we're gonna just identify TMPRSS2 as the serine protease and CATBL as the cysteine protease, just to make it a little bit easier so we're not throwing out a ton of just letters and numbers. And The authors drew on the many similarities between SARS-1 and SARS-2 and the amino acid homology in the spike proteins throughout these studies. And so overall, SARS-1 is used as a foundation to study SARS-2, and this approach was often very effective. So here is a table that we prepared to kind of illustrate the different cell lines that were used.
It is uncertain if the sequence similarities in SARS-1 and SARS-2 viruses translate into similarities in protein function. The lab hypothesized that the SARS-1 and SARS-2 viruses behave similar for viral entry. To better understand the method of entry for these two viruses, the lab decided to investigate the S protein and some of the proteases involved in S protein priming. Because there was no model to study, they first had to set some groundwork. We can see, outlined in red, the protein sequence and incorporated into our pseudotype virus. The immunoblot demonstrated successful expression of the S protein as well. The pseudotype virus is given foreign surface proteins that it is not known to carry, so that these viruses can be given the ability to interact with new proteins. It is used for many reasons: safety, validation, and in this case, to study two different S proteins. The proteolytic response of the 293T cells, shown in red, which suggests that the cell line can cleave the viral spike protein, enabling function of the S1 and S2 subunit of the S protein, which is essential for viral entry. Here we can see, shown using arrows, that both the SARS-S and SARS-2S protein were successfully expressed in the 293T cells and the pseudotype virus they produced. The kilodaltons were very similar, and the faint patterns indicate S protein cleavage, which is expected in both viral proteins. This was evidence indicating that using a pseudotype virus to infect cells may be a good model for comparing these two viruses because the proteins produced still function properly.
Proteolytic processing of the S protein is analyzed by running the samples on an SDS page gel and then immunoblotting to determine if cleavage is similar in SARS-S and SARS-2S due to homology in amino acid sequences. The human cell line 293T are transfected for HA tagged SARS-2S, SARS-S, or empty expression vector, which is used as a negative control. The 293T cell line is used because of its high transfectability. The pseudotype virus will have VSV envelope proteins with the SARS-S and SARS-2S protein. Pseudotype viruses are used to study virus entry, receptor usage, and to safely study viruses due to concern of infecting the researcher. SARS-2S expression and pseudotype incorporation is analyzed using an antibody against the C terminal of HA tag added onto the S protein sequence so it can be translated with the tag. Beta-actin is used as a cell lysate loading control, and VSVM is used as a pseudotype loading control to ensure the same amount of protein is added to each well. VSVM is the VSV virus matrix protein, which is present in all of the samples which use a pseudotype virus.
It had previously been known that replication-deficient VSV particles containing coronavirus S protein could faithfully replicate viral host entry. Experiments done in this lab, shown in the red box, also revealed that both SARS-S and SARS-2S pseudotype viruses would infect the same cell lines with very similar magnitudes. These two pieces of information, we have strong evidence that would indicate that both pseudotype viruses have the same host range, as the arrows demonstrate the cell lines tested. Thus, this would be a good model to compare SARS-CoV and SARS-CoV-2 viral entry. It was next important to identify what genetic similarities translated to functional ones. They examined the conserved residues, shown here in red, and found that SARS-CoV-2 may bind to ACE2, a known surface receptor responsible for SARS-CoV entry.
A broad range of well-studied human and animal origin cell lines are inoculated with pseudotyped VSV expressing SARS-1s or SARS-2s spike protein on its envelope, or VSVG, which is VSV with its glycoprotein, which is used to mediate viral fusion and is a positive control for all experiments done in this paper. The multiple cell lines are used to determine if viral entry was similar between SARS and SARS-2. Pseudotype entry is analyzed by determining luciferase activity in cell lysates by measuring the activity of firefly luciferase in cell lysates. Luciferase activity is used to determine the entry of pseudotyped viruses into the different cell lines. The pseudotype virus contains expression cassettes for eGFP and firefly luciferase instead of the VSV open reading frame. Luciferase proteins are produced when the virus is able to infect the cells and is used to verify viral infection into host cells. Signals obtained for particles bearing no envelope proteins are used for normalization.
BHK21 cells, which were previously shown to be immune to the pseudotype viruses, became susceptible to both pseudotype viruses after the addition of either human or bat ACE2 proteins to the cell surface, as indicated by the box. However, there was no entry of SARS-S or SARS-2S into BHK21 cells expressing the known receptors for the 229E or MERS coronaviruses. Only the ACE2 expressing cells had viral entry. This would be strong evidence indicating that SARS-CoV-2 virus may use the same ACE2 receptor as the SARS-CoV for viral entry.
They did this by inoculating BHK21 cells transiently expressing ACE2 of human or bat origin, human APN, which is the entry receptor used by HCoV229E, or human DPP4, which is the entry receptor used by MERS-CoV, with pseudotype VSV with VSVG, SARS-S, SARS-2S, MERS-S, or 229ES. BHK cells are used since they do not readily express any of the receptors being tested and are not susceptible to infection by any of the viruses being tested. MERS and 229E are also tested since they are coronaviruses which infect humans and are known to enter the cells using a different receptor, which are HAPN and HDPP4 respectively, than SARS-1 or 2, which use ACE2. Cells transfected with antivectors are used as a reference. Transduction efficiency is quantified by measuring the fluorescence activity of firefly luciferase in cell lysates.
See here, indicated by the arrows, that both pseudotype viruses had the same response to the addition of the ACE2 antibody, a dose-dependent blocking of viral entry. BSV and MERS entry was unaffected because they do not enter using the ACE2 receptor, supporting that viral entry in SARS-CoV and SARS-CoV-2 are similar. With this data, it was clear that the ACE2 binding domains on the SARS-CoV and SARS-CoV-2 are responsible for viral entry. With this groundwork set, they could test to see if both SARS-CoV and SARS-CoV-2 S proteins function in the same way.
So far, we know that both SARS-1 and SARS-2 S proteins require interaction with the ACE2 receptor for viral entry into the host cell, and we know that both proteins have similar sequences and function. They also undergo similar cleavage and processing by host proteases.